I just wanted to ask, for the exam on the 28th, will it be covering all the topics or just a few?
Yes, that is your exam, your final exam. Unless it says mock exam, but that too will be on, that will also be on everything. So students normally write 2 exams, a mock exam, and then they write the final exam. So just check the message that you've got. If it's a mock exam, because the normal exam starts between May, June, but it is compulsory for you to do both.
An exam scheduled on the 1st of June, but I'm not sure. I'm just wondering about the one coming up on the 28th.
That will be a mock exam, and that will be on everything also. So it's like a pre-practice exam, almost like your matriculants would write prelims and then the final.
Okay, thank you.
Yes. Students, please do take note that the centers, like your Johannesburg Center or all centers, they have classes in which they prepare you for the examinations, how to use the noodle, what you can expect when you need to pre-register for the exam so that you can get that code that you need to capture. You know, these are very important things because many students don't end up writing the exam because of just technicalities. So please do check with your center and attend those pre-exam classes, those IT classes. It's normally taking place in the labs. However, each center has their respective timetables for these classes. So I know if you're from Johannesburg, the center is also having one on the 29th, Saturday the 29th, I think from 9 to 11. But all the other centers also have it. Just check the one closest to you, and then please attend. It's very important. Unless you have written exams with UNISA before, it can become very overwhelming just getting in to the system, never mind still writing the exam. So please check these out. Thank you.
Today, I'm just going to start by concluding the first theme, which is the themes of biology, with the example. I think we stopped here last week, where it's just a case study in a scientific inquiry. We're talking about how they develop this scientific development and studies, where it's going to be, for example, of this example, when they say a case study is equivalent or similar to an example, but usually when you are writing or when you are doing an experiment, there is what you call it a scientific writing, where you just need to put a distinctive terminology which equates or are equivalent to the scientific format. So a case study in a scientific inquiry, like what Ms. Somaya was saying, usually the word mock exam, it derived from law and all of those things, but it's just a terminology to term it a trial or something like that. So here they will be investigating a code coloration in a mouse population. So for example, here is what they're going to be doing is that the color pattern of animals, as you might or might not know, that they vary widely in nature, sometimes even between the members of the same species. So even the members of the same species, we are people, all of us, we came from the same species, but you will find that there are people who are light in complexion, there are people who are very dark in complexion, there are people who are average and all of that. So there will be variations in those patterns of color in the animals, in the nature. So two population of mice belonging to the same species will therefore just hold up here. There we go. the two populations of mice from which are belonging to the very same species. One species will be Muscus pollinatus, but with different color patterns are found in different environments. So these species, they are found in different environments, but they belong to the same species, but have different colors. And also, they are also in different environments. The beach mouse live in a white sand dunes. So the beach mouse lives on a white sand. This is the environment. The white sand is the environment for the beach, for the beach mouse, and with also the sparse vegetation. The inland one lives in a darker soil kind of environment. So these are the ones, the inland one, this is the beach one. So these little fellows here, they belong to the same species, but they have different colors. We're gonna talk about how their colors is derived from, and also they also live in different environments. You see this fellow here lives in a coast here with a beach population, precisely in the terms of location of the place is the Gulf of Mexico, and this one is the inland population. Here is the beach population, and here is the kind of like you can see here there's a patch which is like a grassland, a semi savanna arid area, and at the edge here you can see that this is the start to be a forest vegetation, or towards your left. And this is this little fellow here, and this is the inland one, which is dark in color. So the two types of match here is the explanation is that they match the correlation of their habitats. That's the reason why this little fella here is white, because it's matching their habit, the correlation of their habitat, and this little fellow here is matching the color of their habitat, which is a bit brown, darkish, if I may put it like that. So natural predators of these mice are all visual hunters. So their adaptation to the color of their environment is derived from the fact that the natural predators of these fellows of these mice are all visual hunters, that they depend on their visuals in order to hunt. So if the color of the mice resemble of that of the environment, they stand a better chances to avoid the predators. So Francis better some hypothesized, we talked about a hypothesis last week, hypothesized that the color patterns had evolved as the adaptation to protect the mice from its predators, which makes sense and which you might find it hard to dispute this hypothesis. So if somebody could just say, why explain the adaptations, reasons why these two mice, which belong to the same species, have different colors. You might actually have to quote Francis Bertoni's summer hypothesis, where he hypothesized that the color patterns of these mice are evolved because of the adaptation to protect themselves from the predators. In 2010, Hoestra, and a group of students tested this hypothesis. So a hypothesis, like we said previously, it has to be tested. So when you are, that's why I didn't like the term guessed, because if you are just guessing, it dispute or it disqualified the test of that hypothesis. So a hypothesis is something that if somebody else who can come 10 years later after you have made your studies or your experiment, can come and repeat the very same hypothesis and actually come out with a result, even if it's not the same results as you got, even it's going to be something else, but they should be able, this is like a hypothesis is supposed to be like a formula, something that you should be able to test. And they did this, and the researchers predicted that the mice that did not match their habitat will be preyed on more heavily than the mice that did match the surrounding. They built these models of mice and painted them to match the ones of the surrounding and place the equal numbers of the type of each model in each habitat, then they record the signs of the predation. So basically here is just that the explanation is very simple in a manner that the mice that does not match the environment will be preyed more or will be eaten more compared to the one that match the environment. So if you come here and put the dark black one, the dark brown into this environment, the predators here, whatever it may be, polar bears or whatever that are here, they will eat this dark brown one compared to this one because this one will be more visible than the other mice, which is the only explanation that these researchers predicted, that they will be preyed more. And this is the kind of experiment that they build, where they build the mice of models and replace them and place them in equal numbers of each type of model in each habitat, and then they recorded the signs of this. And here is what they did in the experimental format. You see in inland, these are the percentage of attacked models. These are the models here, like the mice, the one with light models, the one with dark models. So here on your left is the percentage of attacked from 0 to 100 is the amount which were attacked. So in the beach habitat compared to the inland habitat, you see there are non-conflict and the conflict, which is the control. This is the experimental, and this one is the control. So here you can see in the beach that the one which were attacked, the light one, is just below, just above 25, but the dark models which were attacked, they are even almost over 70, and here in the inland, if or also if you put the light one, you can see how visible they are compared to when they are here. Here you can almost pass it even if you using your naked eyes. So this is the explanation of how different species, they adapt the color of their environment in order to avoid predators, so that they cannot be able they can be able to avoid the predation of their enemies, so that they could survive. So this is just a little fella here, control conflict here as it showed, and here is non-conflict in the experimental, and this is the non-conflict in the experimental. You see how visible it is, and this is conflict in the control. So it's safe here. It's got more chances of surviving than when it is here in the experimental environment. So experimental variables and control in an explanation is that in a control experiment and experimental group, the non-conflict one, which we see on the diagram in the previous slide, is compared with the control group. Yes, the conflict one. An experimental variable are features or quantities that vary in an experiment. That's the explanation of it. So when you talk, usually your lecturer will be talking about what are the variables. Experimental variables are the features or quantities that are the differentiates between in the experiment. The independent variable is the one that is manipulated by the researcher, while the dependent variable is the one which is predicted to be affected in the response. So the dependent variable is the one which is going to be affected, or which is going to be a response to the independent variable, and it's predicted to be affected as a response to the independent variable. So in the contents of science, any question while we are still here, and just watch out for this as an experiment, because if I'm just looking at this and trying to be in the mind of your examiner, he can just, with the case study of a he can give you this diagram here, but it won't show. I don't think they will show these colors. They can just put the diagram with numbers here, few numbers, and they say explain explain the differences between the inland beach habitat or the beach habitat from the format which is here in the diagram. Or they can also just give you the format in the numbers and then say with the figures and say with the representation of a diagram, explain the differences between the inland habitat or the beach habitat with the use of experimental and control variables from the figures which are in the information above. And then you might have to put a diagram like this. So you might need to watch out for this case study as an example that your examiner might extract it and request you to put an explanation on the differences between the same species, two species, which two individuals which belong to the same species but have different colors while inhibiting different environments. What is happening there? They might ask you to explain that with the use of experiment in the form of diagram or in the form of that kind of explanation. So theories in science, in the context of science, theory is a broader scope than hypothesis because in general and can lead to testable hypothesis, yes, which is supported by a large body of evidence in comparison to hypothesis. So the difference between a theory and a hypothesis is that a hypothesis, you don't necessarily need to have an evidence to prove your hypothesis. You just need to have an educated guess to what is going to happen if you test your hypothesis. But a theory in contrast have to be backed by an evidence, and it have to include a test hypothesis which has been testable and which has been proven by a large body of evidence. Then science benefit from cooperative approach and diversity points. Most scientists work in teams, and which often include graduate and undergrad students, which are you guys. And teamwork always thrives. So if you are working, you might actually need to form and find somebody who is doing the same thing as yourself. Then you might be able to get an assistant and get a help in some form or the other in your work. Then good communication, just like in anything, is important in order to share results through seminars, publication, and websites. So usually when you are doing your honors, your MSc, yeah, perhaps, and even on the undergrad, yeah, you will have to go through what they call seminars and publications. Excuse me. Publication is like we talk about this experiment. After the result that they will get here from this experiment that they were doing, they can go and do what you call a publication. When you do a publication where you present, you publish your results. What did you find? What does this mean? What does this mean? What does that mean in the comparing when you're comparing the inland mice compared to the beach mice? And then the other person who will do the similar thing that you did will have to coach you or will have to reference you, and then you've got publication, and then with those publication you can be rated in institutions like science publication and NRF and all of those. So you can also do publication on the website and do what also presentation on the colloquiums and in the events of the institution where you're going to have to present your work about the results that you found in your experiments. Building on the work of other scientists, check each other's claims by performing similar experiments. So what you're going to do, if you're going to do an experiment, others can come and do it. and increase on it, or disprove on it and say, actually, and it does not mean that what you did is wrong or you were lying or anything like that. It's just that science is broad and there's a room for science for anything. That is why the main point in science is that when you do a research, your hypothesis, what you're doing, somebody else has to come and do it later. And people can disprove your theory or your results, not because you were wrong, just because the variables has changed, environment has changed, because now the dynamics of that environment are no longer the same, just because the factors such as rainfall, like if you did an experiment 10 years ago and somebody come and do it 10 years later, now 10 years later, the amount of rainfall is not as frequent as it was 10 years later. Now there are new species. If you're dealing with a species, now there's an invasive species plant who has inhibited the new in the habitat is now a new territory at all. And you can find, if you are dealing with water scientists, now there are new variables. The streams are no longer the same as they are, because the end of the stream, where if those streams used to end up, if the stream used to end with to a bigger river or to a dam, now that dam has been disturbed, or now at the end of the stream the demand is very high. There are a lot of people who are in the receiving end of that stream. Now the flow of the stream is no longer the same as it was 10 years ago. So the fact that the new people has come and do and disprove your experiment, it does not necessarily mean that the person who did the experiment before was wrong, or that. It's just that the procedure is that this scientists check each other's claim by performing similar experiment, and if experimental results are not repeatable, the original claim will have to be reserved. Like what I was saying before, it is not unusual for different scientists to work on the same research question. It's not. It's very, 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 very. It's just that the people who are very maybe innovative, you will find that if you have a research topic which is similar to somebody else, not similar per se to word, but if you are just the objective are similar, you wanna check the micro or habitats which are in the microorganism which are in the certain river, or you wanna check the different type of plant species which are in the park in Pretoria, you wanna check, and somebody else has done that before. So people who are innovative will be like, why are you doing something like that which has been done before? But it is not unusual for different scientists to work on the same research questions that have been worked on before. Scientists cooperate by sharing data about model of organism. For example, the fruit fly, the Dispolia, Melegostana. So here, when they mean that they cooperate by sharing data about the model of organism, there could also be information. Like if you, for example, you go and collect the how many mice are there in the beach habitat of, let's say, of the one for Mexico that they use an example. So that data will be recorded. So somebody else who is doing a write-up about the mice of a different region of Canada, let's say, of other environment which is near Mexico, can include the data of Mexico by just quoting or referencing that the previous studies by UNISA et al., UNISA and so have indicated that there are how many number of species of mice which are in the beach environment of Mexico. So that's what they mean, that you can cooperate by sharing the data, although the most important thing, like what we said when we're doing the introduction, is to reference, because if you don't reference, you will be classified as plagiarism, because you actually need to reference to show that this is not your work. You cannot, if those studies, those people find that there are 20 mice in Mexico, beach of Mexico, and you go and just say, I know that there are 20 mice of Mexico. The question will be, how do you know that? You need to reference and say, according to a study by so and so, there are 20 mice in Mexico in 2023. So, which means this and this and this. This is what they mean by cooperate, that they cooperate and share the data of the model of organism. The science, technology, and society. The goal of science is to understand the natural phenomena. The goal of technology is to apply the scientific knowledge for some specific to the purpose. So therefore, biology is marked by discoveries, while technology is marked by inventions. So that's technological. In terms of technological work, these people are always inventing new inventions where new techniques and new mechanisms and new strategies or new materials in which we can be able to understand the discoveries which we discover in terms of biology. So my old professor used to say, you will never discover anything if you're in your office or in your lab. So, which means I know there's a group of people. One of my friends is a lecturer here in your institution. They're in Durban as we speak, in the Department of Environmental Science, and we were talking about this, that you discover more if you're gonna the chances that you're gonna discover new species is when you're out in the field. So when you go and say, I'm going to check how many, the count of red deer, the count of elephant shrew, the kind of how many species are there in La Juma, in whatever nature reserve or whatever in Musina, in those areas, there are big projects where, like, for example, if you are in a, not if you are in a, there will be a new developments. Like I know of a project which is in the Musina. Musina is just a town before the border of Zimbabwe and South Africa Bed Bridge. I'm sure most of you know about that. So there are this type of investors who wanna, quote unquote, bring some development and put an investment of a lot of money. So they wanna develop that place or area, which is literally, if you've been from in the N1 from Lois Richard to Messina, it's just a long 2 hours of drive where you are just passing. Both your left and right is just bushes and just wild. So they want to invest in that area and start a development where they're going to build mines, where they're going to build shops, they're going to build schools and everything. There is, it's a big project which has got a lot of money. And so what they do before they could approve that, before the government or the municipality they can approve that, they'll bring you, UNISA students, and say, if we clear out that area, how many species are there? How many species will be affected? So you are the one, when you go to that area and assess that area, there will be people who are specialists, plant, the one who's going to be assessing the environment in terms of, with the view of plants. They will be saying, yes, those plant species that are there and there and there, are how many plant species are only endemic to that area only, that if we clear that area, those species will never be anywhere. Like there are type of tree species which are only endemic to one specific area, that even if you attempt to plant them in area which is just maybe a few kilometers away, they will not grow. And other other animal species which are there, which you can advocate that if these animals are cleared out here, if you clear the environment, therefore these species will be they will not recover. You will not find them anymore. So how do you discover that? When you are in the field, that's when you stand a big chance of discovering a new thing in terms of biology. Then technology, like people who are doing bioinformatics, you can sit on your lab, You can sit using your models, using your models such as R softwares that you just need data and information or a data. How many plant species are elsewhere? How many inputs are elsewhere? Then you can be able to manipulate, not just necessarily manipulate, but you can be able to put some readings which could translate to meanings, which could translate to the data information, which could be published and be able to predict what is going to be the future like, or what the trajectory of that particular species is going to be like in the near future. The combination of science and technology has dramatic effects also on the society. For example, the discovery of DNA by James Watson and Francis Crick allow for the advances in DNA technology such as testing for hereditary diseases. Yes, you can, by the means of DNA, you can be able to detect the diseases that you have inherited from your parents or your relatives. Debates on technology center more on should we do it than can we do it. That's those kind of debates that are for in the in where if you are looking at into in terms of technology. Ethical issues can arise from new technology, but have as much to do with with politics, economics, and cultural values as with science and technology. There's always politics in everything, so I think that is self-explanatory. Then the value of diverse viewpoints in science is that many important inventions have occurred where different cultures and ideas mix. For example, the printing of press relied on innovation from China and Europe. So there's a mix between the culture and technology and also ideas. For example, if you're gonna be conducting experiment and you're gonna need to collect your data from remote areas. By remote areas, I wanna say like from rural areas. There's gonna have to be that mix of culture. There's gonna have to be that mix of collaboration where, in case if you wanna collect your data or you wanna conduct an experiment or a survey in areas, you're gonna have to put into consideration the views of the villages, the views of people in those areas, the views of culture. How do they mix in terms of indigenous knowledge? In some areas, they can tell you they have an indigenous knowledge where they could predict where is where is it gonna rain. In some areas, they can predict how the biodiversity or the vegetation is going to rejuvenate, is gonna recover, because you know those areas there are people who also clear the bushes or the grasses, you know, in rural areas where we come from. They used to make roofing with grasses. Does anybody know about that? Just show by your thumb if you know about that, where they used to make roofing with grasses and all of that. Nobody, yeah, okay. Some people know. Yeah, so there's a bit of an indigenous knowledge that needs to mix with the new ideas. So science benefit from diversity from different racial and ethnic groups, from both women and men. Like for example, I'm conducting a research study on Mopane worms. So in ancient history or previously, long ago, but like 30 years or 20 years ago, the people who used to harvest the Mopane worm were just women and all women, all women, yeah. Men and young men didn't bother about them. They could eat the Mopane, but the collection of them, there was a stereotype kind of event. But now as times goes on, obviously, when there's they talk about economics, the economics, now the politics, but more importantly, the economics, you know, things are expensive now in our country. Now even men and young men go and harvest and remove that stereotype and collect those Mopane worm, and they go and do the trade. But although there was also an a view that it's not necessarily a view, but it's just a indigenous knowledge that if they dry those mopane, because when they harvest them, they are still alive, but they will collect them and preserve them. Then they will dispose the insides of the mopane when they will then dry them. And when they dry them after like a week or so, when they are cranky and so forth, to use the young children language, they will crush them. And they crush them, they will do what they if you are using your blender to crush them, but because in the ancient history or back in or in the rural areas they don't have the blenders, they'll just crush them using those sticks, not stones, and then they will mix it with a soup, and then they will give it to young kids which are suffering, or even individual adults who are sick from those kwashiorkor or those diseases which are caused by malnutrition or undernutrition, and they will recover easily. And it has been a remedy which works very well. So it's an indigenous knowledge or a cultural knowledge which can mix with science, and that's where science benefits from the diverse views from different racial and ethnic groups. The more voices heard, the more robust, valuable, and productive scientific interchange that the more there's differences of ideas, the more there will be a robust arguments, not in just a form of people are arguing. You're like, hey, no, this, yes, no, not like that. If people are publishing, if people are coming with more information about the certain view, about the certain plant species, about the certain animal, about the certain view, they're more productive. There will be a scientific interchange which will lead to more and more discoveries. So for example, here, if you just want to take so here, this, if I put this figure which is published in the UN 002, so is this is, what is this? Is this information which is here, if I can just put a question, because that's also one of the good questions that you can be asked. What is this? Is this heading? How will you describe this heading as? I think even the word heading is wrong. Anybody who can go? Okay, I know my question is wrong. Let me just give it easy for you. Is this a title or is this a figure? You can type or you can just, but the quicker one will be just to unmute yourself and say something. Anyone? Is this a title or is it a figure? It's either one or it's either or. So scientifically, if you're putting it like this, if your title, if your heading or whatever that you are describing is at the bottom, it will be described as a figure. So if you come here and put on a title and say this is a title, before you put data from D.W. Kaufman adaptive coloration in the Prescriptor pollinators experimental selection by all Journal of Mammal from chapter 55, pages 271 up to 283, 1974, and put it as a title. Then if your examiner is not in a good mood, they might wrong. But if it's at the top, your title, your description is on the top, that's why it could be described as a title. So here you can see that there is a differences between number of mice caught and number of mice caught here. But what is the difference? The difference is that it's a light colored soil, the dark colored soil, and one of the variables which are different, this is a full moon and no moon season. Full moon and no moon season. So when there's a full moon on a light colored soil, you can see that the number of mice which are caught, they are, yes, very similar. That's just below 20. And the number of light coat mice which are caught, which are which are catched during the no moon season, it's very low compared to the dark one. But when there are full moon, you can see the differences that the number of light caught, which are caught, are very higher compared to the dark ones, and so forth even here. So the difference here is that the light one, they are more which are going to be caught. The dark one, there are more which is going to be caught when in a no moon season compared to the one in a full moon season. I don't know if this figure is making sense. So the first one is a light colored soil. This is on a light colored soil. The second one is on a dark colored soil. So the previous one, it was on the dark on an inland, but the other one was on the beach. So which is still the same, because this is just a light colored soil. The colored soil for the beach, yes, the dark colored soil. So when there's a full moon in the light colored soil, you can see that yes, the dark one, the dark coat, or can be more visible compared to the full moon. There you can still see it, but when there is no moon, there is no light. That's the understanding, or that's the common understanding. So when there's no light, so the description, or we're gonna be depending on also on the visibility of the individuals. So this one will be the dark one will be more visible because there is no full moon, there's no moon which is showing, and the one which is light will not be extremely visible. But in the dark colored soil, when there's full moon, the light mice is very visible. You can check. And even if there's no moon, because the soil is dark and this mice is very light, it's also visible and like that. So you can be able to distinguish how these structures and the experiment could move light. So this is the predator there, which is going to be coming either when there's full moon or no moon in the light colored soil. Should also be coming in the full moon in the dark colored soil, which is going to be coming on the day where there's no moon, and on this one. And how will it see? And it will be able to see and be able to detect and see how they could be able to prey on their prey. But the general assumption, or the general result, I don't want to use the word assumption, the general result is that the general results is not that the light mice is more susceptible for predators than the dark coat mice. No, the general result is that the ones which resemble the color of their environment is less susceptible to the predators. So if you are an individual and your color is opposite to your environment, you are more in danger than those who have resemble or have the adaptations of the color of their environment. That's the general results that the data gives. This we did on the first slide. Remember the energy flow, where it's going to be coming from the sunlight, and then it's going to be going, and then it's going to be going for individual organism, and then there will be energy flow in a form of chemical cycling. We also did this. We also did about also the beakers, about how they are adapted to their feeding, and how these two have different beakers, because this one feeds on depending on what they feed on. Some feed on seeds, some feed on insects, and the adaptation of the beakers was determined by what they feed on. So this is just a diagram of the population of organisms where there are hereditary variation traits that you have inherited from your parent species, which is going to be passed on to to your children or to your offsprings. Then there's overproduction of offspring and competition, which then is differentiated by environmental factors. And then there's another level of differences in reproductive success of individuals. Then there are evolution of adaptation in the population. So we're gonna go to chapter 2, which is atoms and molecules. I don't know if I'm teaching about something which you are already ahead of. Let me just do this. Chemical connection to biology is that the definition here is that biology is the study of life. Yeah, we discovered that, or we noted that in a previous sessions. Then organism and their environment are subject to basic law of, here we're going to be talking about a little bit of individual practice, what called physics and chemistry, and you will understand as we go further what do we mean by this. One example is the use of anybody want to say something? Yes, unmuted yourself by mistake there. Okay. One example is the use of formic acid by ants to protect themselves against predators and their microbial parasites. So they release this acid in order to use it as a form of shield to shield themselves against their predators and their microbial parasites and all that. So this formic acid will be able to help them, like I've said, against their predators, these little ants. There are thousands and thousands of different species of ants. I know somebody who's doing the study about ants that there is almost infinite about the thing that ants represents. I know that person started doing their research on their honors level all the way to their MSc. They finished their doctoral studies. The subject point as ants with a survey on the and we're doing a lot of experiments. He is still doing even today. He's now a lecturer at University of KwaZulu-Natal and still doing this. There are a lot of thousands of species and a lot of developments and case and points that you can discover using ants as a point of case and study in terms of genetics, in terms of ecology, in terms of conservation, in terms of population dynamics, of environmental structure and spatiality of those structures, which are determined by ants. And you will be very, 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 very surprised that these little tiny individuals who seems very useless and no point at all in the society, but they contain a basic or baseline of the environment at some point. And this is just an individual with a graphic representation. Then concept 2.1, that the matter consists of chemical elements in a pure form and in combination called compounds. So you might want to do that and use that as a definition there of a compound. Organisms are composed of matter, where matter is anything that takes up the space and has a mass. So in the elements and compounds, this is just a description. We're going to be reading a lot here. These are just some notes that you're going to have to very pay extra attention in order to deduce and be on the lookout for the words that your examiner can ask you to describe or explain or define, as it were. Matter is made up of element. What is an element? It's a substitute that cannot be broken down into other substance by chemical reaction. And the compound, what is a compound? Is a substance consisting of two or more elements in a fixed ratio. A compound also has a characteristic which different from those of its element. Then, if you can check here in the first diagram representation is sodium, which is combined with chlorine. Then you get what you call a sodium chlorine. Why does it look like this? You know what? This little fella here. This little fella. What is it commonly known as? You can use you are allowed to use your Google, I think. Yes.
Table salt.
Good. Very well. Good. That's sodium chloride table salt or just salt. Yeah. The elements of life is about 20 to 25% of those 92 natural elements are required for essential elements, where those, for example, are carbon, hydrogen, oxygen, and nitrogen, which make up a 96 of living matter. The most of this remaining 4% out of this 96 consists of calcium, phosphorus, potassium, and sulfur. So your examiner can ask you what name elements, essential elements, which comprise of 96% of the living matter, and where you might have to describe those as carbon, hydrogen, oxygen, and nitrogen, and where the remaining 4% are calcium, phosphorus, potassium, and sulfur. Trace elements are required by an organism in only a minute of quantities. So we'll explain what does that means when we go further. Then this table just describes the elements in the human body, where if you can, so the other thing that you might need to look out for, I know somebody might say, it's not as important as anybody could wonder about. So these are elements. This is a symbol you need to catch up for. The Ca, which is stand for calcium, and the P, which stand for phosphorus, and the K, which stand for potassium, then sulfur, and sodium, then chlorine and magnesium, and those are their percentage. Then 96.3, which make that 96 that we talk about, and therefore 93.7, which will be converted to 4%, which means this 4% that we talked about here. And if you can check, those are the ones that we talk, that trace elements less than 0.1%, which are boron, chromium, cobalt, copper, and fluorine, iodine, iron, manganese, and other. You can see that they are the ones which have less than 0.01% of the mass, which are of the elements in the human body. And this percentage, in case when they wanna just describe, they also include water, and sometimes they could say percentage of which are included in the water. So these are body mass, but also it includes the water in them. That's why they are also converted into the nearest. Then the case study again is the evolution of tolerance to toxic elements, where some elements can be regarded as toxic, and some elements can, where some species can become adapted to the environments containing toxic elements. It's not anything other than what we're talking about. It's just that here, it's just that there is a descriptive of the danger in which those species which have to be adapted to the environment, are adapted to the environment using the resin as such. Therefore, for example, some plant communities are adapted to serpentine of their environment, and where you can see if the adjusted year of this plant species have to be adjusted and adapted to these communities of this environment. By communities is that they're including everything which is part of this environment. So the rocks, the shape of the environment, you can see here. I know they didn't describe this, but this always an uphill kind of environment which is not even which is not in an even format. There are other some tree species which are further apart from there. I'm sure if you can move further to your left down, there could be also a stream where there are like sedges, plantains, and other species. So this plant species have to be adapted to this planet of this community. And all that you see even here, there's a description of a little barrow here of a road, which is forming also an edge and like that. And if you make something like this in an environment, plant species, you have disrupted their movement and their distribution in a more bigger format than you can imagine. You might think that there's still a little bit chunk of environment on either side, but you have put on a barrier which maybe some of them can differentiate themselves into their dynamics of their vegetation. And this is the plant and this is the pennant of that we were talking about. Then 2.2, where we talk about elements properties that depend on the structure of each atom. Excuse me. Each element consists of a unique atom. Yes. What is an atom? Which is the smallest unit of matter that still retains the properties of an element. Then in those atoms, there are subatomic particles where atoms are composed of. The atoms are composed of subatomic particles. Relevant subatomic particles includes neutrons, protons, and electrons. Neutrons, protons, and electrons. Neutrons, they are not electrically charged. Protons are positive charged. Electrons are negatively charged. So you're gonna have to get that and be able to hold on to that. So neutrons and protons form what we known as atomic nucleus. Remember, you're gonna have to hold on to this. Neutrons, they have no electrical charge, and protons, they are positively charged, and electrons are negatively charged. So neutrons and protons, the ones which has no electrical charge, and the one which are positively charged, they form an atomic nucleus. Electrons form a cloud of negative charge around this nucleus, where now the neutron's mass and the proton mass are almost identical. And therefore, the unit measure for those is in daltons. So if you want to measure the neutron's mass and the proton mass, you use it, and they're measured by the unit called dalton. Then here is just a representative in the form of diagram, where in A you find the cloud of negatively charged 2 electrons. You see these electrons, they are charged by electrons which are charged negatively. So if you can just see here with the description, and inside here you can find that there are nucleus which are inside, and then they form these 2 little positive charges here over here. But these electrons are electrons. They are cloud of negative charge, which are true electrons. We will talk about this, and you will see it when we describe the atomic numbers and all of this when we go forth. Then atomic number and atomic mass is that they are here again, like what we did when we were talking in the sections in the second session last week. You're gonna have to, I know it can get like very overwhelming and like it's too much with a lot of words, but this is just gonna have to follow up the words now and the description of the words. Atomic number and atomic mass. Atoms of the various elements differ in number of subatomic particles. Then an element's atomic number is the number of protons in its nucleus. So atomic number is the number of protons, in its nucleus. And elements, the mass number. What is the mass number? The mass explains itself, is the weight, is the mass, is the heaviness of the matter of the element, is the sum of protons plus neutrons in the same nucleus. The atomic mass, and I see a lot of people have a struggle to differentiate between mass number and atomic mass. Is the atom's total mass, which can be approximated by what? By the mass number. I know this word seems very similar and so confusing when you're just grabbing them at the same time. But let's continue. We'll make it work. I promise you. Then isotopes are all atoms of an element have the same number of protons, but may differ in the number of neutrons. Why am I saying this? You might have to follow them thoroughly. This was because I see also in your past question papers, in your assignment, in your exercises, there are a lot of questions about the giving the one word terminology, and some other times where you're going to have to describe the differences between protons and electrons, where you're going to have to name what are isotopes. And the examiner, when he gives the one word items, they can sound very, very, very similar, and in turn can be very, very confusing. So you might actually have to watch out for that. So atoms of an element have the same number of protons but may differ from neutrons, and those isotopes are two atoms of the same element that differ in number of neutrons. So here isotopes are two atoms of an element that differ in number of neutrons. But the biggest description here is that isotopes are two atoms. There are two atoms. That's a term that you need to, because if there's something else other than atoms of an element that differ in the number of neutrons, it's no longer an isotope. A radioactive isotope decays spontaneously, giving off particles and energy. You don't have to worry more about that, but it's just a description of radioactive isotope. Then there's what we call radioactive tracers. Radioactive isotopes are often used as a diagnostic tools in medicine. I remember in the last year we did a question like this, where they asked something. It's just that I forgot, but we will see it when we go to the point where we're going to be dealing with past question papers, where they ask you to, either they ask you guys to describe how a radioactive importance with regards to everyday life or in our day-to-day lives, or how important are radioactive traces. Then in one of the things or the question or in memorandum, you had to describe that radioactive isotopes are often used as diagnostic tools in medicine, and radioactive tracers can be used to track atoms through metabolism, and they can also be used in a combination with sophisticated imaging instrument. So these are where they use radioactive when they are doing some diagnosis into maybe to check if somebody has a tissue of cancer in the throat and all that. So another format which they could describe a question, they could just say, give the function or the function of radioactive tracers. You will have to describe this because the uses are function. When you talk about some, if they are describing something as it's used for, it's used to track atoms through metabolism. I often used as diagnostic tools in medicine. They can also be used in combination of sophisticated imaging instrument. These are functions. This is not what radioactive tracers are, but these are function of radioactive tracers. So if they ask you about describe what is the radioactive tracers, the functions of the uses, or what are the radioactive tracers are used for, describe how or what radioactive tracers are used for, you will have to point out these functions. And already if you put 3 as they are in the distinctive format, it's already 6 marks that I can guarantee you that you have it in the back. So also this, we talked about this cancerous throat tissue. So radiometric dating, a parent isotope decays into its daughter isotope at a fixed rate. A fixed rate, you know what fixed rate is. A fixed rate is it's a rate which is here they express it, or they describe it as the half-life. And also I'll come back to explain what a fixed rate is. In radiometric dating, scientists measure the ratio of different isotopes and calculate how many half-lives, what are half-life, with this current isotopes, the rate in which this current isotope decays into its daughter isotope in this fixed rate, which is expressed as a half rate. So in radiometric dating, scientists measure the ratio, not radio, sorry, the ratio of different isotopes and calculate how many half-life have passed since the fossil of rock was formed. So half-life values vary from seconds or days to billions of years. So there will be fossils which are going to be described previously from previous billions and billions of years ago, where they're going to be described as a half of life. So by fixed rate, they categorize it into saying a parent rate of the parent isotope decays into its daughter's isotopes at a fixed rate. By fixed, they just you know what a fixed rate is. It's an amount which cannot fluctuate. If it cannot be reduced and it cannot be increased depending on the amount of the pressures of the ratings and whatever. So that's how they measure this radiometric dating. And also, so in these electrons, there's what they call energy level. These energy levels of electrons, you know what is the expression of, or the definition of energy is the capacity to do work, or the capacity to change, to cause change. Yes. Potential energy is the energy that matter has because of its location or structure. The potential, the one that can be produced in that environment or in that location. So electrons of an atom differ in the amounts of potential energy, and electron states of potential energy is called energy level or electron shell. So if they request you to describe or define what is an electron shell, because I don't think they will say what is defined, what is an electron shell or energy level. They might just ask you one or the other. So you might have to, you will have to definitely know that energy level or electron shell, they're referring to the same thing. And some other times they will just be giving you, if let's say for instance it's a one word question where you're gonna have to describe one term which describe the definition, where you're gonna have to place an electron states of potential energy, which is called either electron shell or an energy level, because these electrons of an atom, they differ in the potential of this energy level or the potential energy from the energy that matter has because of its location or their structure. So this figure here where the ball is bouncing down to flight of the stairs, which can come to rest only on each step, not between step. So the atomic nucleus here, the first shell is the lowest energy level, the second shell is the highest energy level, the third shell is the higher level, the second shell rather, sorry, is the higher energy level, the third shell is the highest energy level, where here in between the energy which is absorbed and the energy which is lost. And when we talk about the low energy level, we're talking about electron shell. So that's what you can see even here. So this example where the ball is bouncing down a flight of stairs and can come to rest only on each step, not between step. On each step, they bounce once and then they have to go down to the last step. Then electron distribution and chemical properties will then, the chemical behavior of an atom will be determined by the distribution of electrons in an electron shell. In an electron shell, what is an electron shell? Remember this, in an energy level. So these are the kind of terms that you might actually have to underline even when you're studying in your notes, and make a little bit of a note there that when they're talking about an electron shell, what it is that they're talking about. The periodic table, you know about the periodic table of the elements, show that the electron distribution for each element. Then here you see when you're talking about in the periodic table, here they show you you don't have to worry much about this though. They talk about the first shell, second shell. This is the first shell, second shell, and the third shell. You can, okay, let's start with the first shell. You can see the description of a hydrogen, and then there's a description of a helium, where there's also the description of where you can see where is the atomic number, where is the element of the symbol, where is the electron distribution in the diagram, and where you can see the atomic mass, what is this number at the top. You can also see the second shell, which comprise of neon, fluorine, oxygen, nitrogen, carbon, boron, beryllium, and lithium. And the third shell you can see is comprised of magnesium, aluminium, sodium, silicon, phosphorus, sulfur, chlorine, and argon. And here is the description of what these elements are. If you can also make in your notes an example like this in order to check what is which and which is which. So here in the terms of helium, you can see that the top number is regarded as the atomic number, and then here is the element of a symbol, which is He, which is stand for helium. Then the bottom number is an atomic mass. And here you can see that they are saying atomic number, and then there's electronic distribution of that particular diagram, which is also we will introspect that even more later. Then first shell, like we said here, is considered, it consists of helium and hydrogen. So helium, hydrogen, you can see that it's got the one. What did they say this is? These are two atomic numbers. And this one for forgive my voice. For hydrogen, you can say it's 1, and for helium, it's 2. Then so it is also on the second shell, where you can see that lithium has 3, beryllium has 4, then the boron have 4, then 4 has 5 and 6. And even also in the third shell, you can even check how many they are. And then when we go further with this, when we are provoking questions here, you're gonna have to use a little bit of your calculators to calculate the atomic number and atomic mass as we go further, in order to calculate how much is the atomic number and how much is the atom electrical charge, and all of those we'll see when we go on. Then the second shell, the third shell, as it is there in the description. So also when we go further, there's what we call valence electrons. Valence electrons, which are those in the outermost shell, or valence shell, outer shell of valence shell. So the inherent explanation here is that the chemical behavior of an atom is mostly determined by the valence electrons, and the elements with full valence shells are what they call chemically inert. So elements with a full valence shells are they describe them as they are chemically inert, and electrons orbitals. An orbital is the three-dimensional space where an electrons can be found almost for more than 90% of the time. That's where it will be found. Those electrons will be found in an orbital, orbital of electrons. Each electron shell consists of a specific number of orbitals, because these orbitals are the ones that they are saying that 90% of the time you will find an electron in. So each electron shell consists of a specific number of orbital. Then this is what we're talking about now. So here there are three diagrams. There's A, B, and C. So A, we are describing the distribution, electron distribution, electron by itself. Then B, these are separate electrons on their orbitals. Then C is the superimposed electrons or orbitals. So here, if you can just check here, this is with a neon as a subject electron, where neon with two field shells, where there's 10 electrons, 10 electron, 2, 4, 6, 8, 10. Am I right? Correct? Yes. My 2, 4, 6, 8, 10. Yeah. Then this will be the first shell. The first shell will be this more or less like a circle, for a lack of a better word. The second shell will be this one, and then there's a neon with two filled shells. They're filled with how many electrons? They're filled with 10 electrons. And in the first shell, how many electrons are there? They've got 2 electrons in the first shell, and in the second shell, they've got 2, 4, 6, 8 in the second shell. And there's also, when we go further, when we're gonna go about the balancing of this kind of things, where they will explain, your lecturer will be explaining how do they mean by electronically balanced, electrically charged, and all of this. Then the second one will be the separate electron orbitals. Now they are separated here. Then there's a first orbital, which is the first shell, and then there's a second orbital with the second. So the first one, second one with the 2s orbital with 3 2p orbitals, which is explained here. You can see here, even with the X, Y, Z, on these 3 2p orbitals. And then there's in the superimposed electrons, where there's 1s, 2s, and 2p's. But we'll explain it with the form of another example with virtual and numbers, where we can be able to calculate that as we go further. So the first one in the A, where we talk about the electron distribution, that's where when we explain that electron distribution of this electrons of the element of neon, or neon if you wanna pronounce it like that. Then we explain that there are 2 shells and two electrons in the first shell, and then there is a separate electrons orbital in the first shell, where there's 1s, there's 2s, and there's 3s. But the 1s is combined or is separate in the first shell, and the 2s and the 2p orbitals are in the second shell, all combined together. We will also describe and explain what this means in details as we go further. And there's the combination of the superimposed electrons orbital, where there's 1s, 2s, and 2p on the superimposed within those electrons orbitals. Then the concept 2.3, the information, the formation and functioning of molecules, which depend on chemical bonding, between atoms. So atoms with incomplete valence shell can share or transfer can transfer their valence with certain other atoms. Why? This interaction usually results in atoms staying close together, which are held by attraction called chemical bonds. These are the bonds which are the ones that attract this. So what are chemical bonds? Chemical bonds will be attractions, which usually results in atoms that will be staying together, which are held by this attraction called chemical bond. There are various reasons why these are held together, but we will go together with when we go forth and see how these chemical bonds, what come of these chemical bonds, what are the examples, and why are they called chemical bonds, and what are the other inputs or the other outliner, such as ionic bonds and all of these. So other bonds is covalent bond. A covalent bond is the sharing of a pair of valence electrons by two atoms. Remember when we talked here, we talked about the fact that atoms with incomplete valence bond. You might have to underline the word incomplete valence shell can share or transfer valence electron with certain other atoms. Then covalent bond is the sharing of a pair of valence electrons by two atoms. So in a covalent bond, the shared electrons count as part of each atom's valence shell. So this covalent bond, those electrons which are counted there as part of each valence electrons are in a covalent bond. Then, for example, if you are using hydrogen atoms, you can see that these are two positively charged. There we go. Then we go down further with an arrow there. Then you got the hydrogen molecules there. And a molecule consists of two or more atoms which are held together by a covalent bond. A single covalent bond or a single bond, single covalent bond or single bond is the sharing of one pair of valence electrons. So if I said a single covalent bond or a single bond is a sharing of one pair of electrons, I'm wrong, because you need to describe it as of a valence bond. That's why I'm saying when you're studying this, you might actually have to pay extra attention to details of the description. That if you miss one or you omit one stretch of information, you are completely saying something which is different to what you are saying. Then a double covalent bond now, or a double bond, is the sharing of two pairs of valence electrons, not sharing of two valence electrons, sharing of two pairs. There, when we're talking about the single covalent bond, is sharing of one pair of valence bond. I've seen some of the students make mistakes in previous, when your lecturer will penalize you. You find that you omit the fact that you just say it's a single bond or single covalent. Listen to me, I want to say covenant. A single covalent bond or a single bond is the sharing of one of the valence electrons. It sounds like it's similar or it's the same, but it's not, because there's a difference between one pair of valence electrons and just one of valence electron. So you might have to also pay attention to details of such. And even here, when you say two pairs of valence electrons, not two of valence electrons, but a pair rather than anything else. The notation used to represent, notation is just a description or the symbol to represent atom and bonding is called a structural formula. For this, for example, this is a bond where you can check here for hydrogen and check this a little bit. You can call it a dash and all that. Then can be abbreviated for further with a molecular formula, for example, H2. Then this is this figure here where they're going to be describing the name of a molecular formula, where they talk here that the annotation of the notation of the represent the bonding is called a structural formula, which can also be abbreviated as a molecular formula. So there's a difference between molecular formula and also a structural formula. There is the example. So the notation which represents the atoms and the bonding is called a structural formula. Structural formula represents the atoms, and their bonding. But the molecular formula is their abbreviation, which are used for further formula. And the molecular and also a formula electron distribution diagram, then that's what we call Lewis dot structure and structural formula. Then there's a space filling model. You don't need to worry about this though, but I think what you might have to worry more about and look out for is how many electrical charge electrons are in the orbit or in a shell. Then you can check here is water, H2O, or the methane, CH4, oxygen, and all that. And the bonds are also, you not need much to worry about that, but you have to understand them and look out for them into how is their structural formula. It's about the bond here is more like this. So here is so here there's a double bond. Here you can see and the H2O when there are 2 hydrogen and 1 oxygen, and here you can see that there are 4 hydrogen and 1 carbon. See how they put the bonds there, and there's a single bond, and here there's a double bond when there are 2 oxygen, and then this one here. So you might actually, but what more you need to worry more about, and you need to know when you're studying, know when you're gonna be prepared in your exam, is about their formula and molecular formula, where hydrogen is H2, oxygen is O2, water, methane, and other elements which are not here, which they can put as a form. And here that's exactly what we're talking about here in a data and the more microscopic format. They will show you in your notes and all of those, and then they will describe it like that. So bonding capacity. Bonding capacity is called the atom's valence. So covalent bond can form between atoms of the same element or atoms of different elements. A compound is combination of 2 or more different atoms. Just hold up a minute. Just give me a second. There's an audio which is playing here. I don't understand. What is that? Okay. Then sorry about that. The covalent bond can form between atoms of the same element or atoms of different elements, and the compound is a combination of 2 or more different elements. Then atoms in a different atoms in a molecule attracts electron varying in different in degrees, varying in different degrees or varying in different forms. So electronegativity is an atom attraction for the electrons in a covalent bond, and the more electronegativity an atom is, the more strongly it will pull shared electron towards itself. So this electronegativity of an atom's attraction for the electron in a covalent bond will be equipped more if the more electronegativity of an atom is. That's when the more strongly it will pull its the shared electron towards itself, and then that's where the strength of its bond will be towards. So in a non-polar covalent bond, so these are the unfortunate for your studying purposes. These are the, I know it's a lot of terms and a lot of things to grab at the same time or at the short space of time, but this chapter will include a hell lot of this where you're gonna have to to be glued more in your notes and be able to grab all of these terms. So in a non-polar covalent bond, the atoms share the electrons equally. That's in a non-polar covalent bond. But the good thing, I'm sure if you might have noticed about this chapter, is that it's a comparison. It's a comparison. It's a comparison of terminologies. Like for instance, here we're talking about polar and non-polar. You just know that if you understand what is a polar, non-polar covalent bond is, if you understand what a polar covalent bond is, then in turn you have understood what is a non-polar covalent bond. And the good part is that your examiner, when he's going to be examining you, you want to understand, you will want to get your understanding in terms of the difference between this terminology. Like here, you checking if you understand that a non-polar covalent bond, the atoms are shared equally. Then contrary to that, polar covalent bond, one atom is more electron negative, and the atom do not share the electrons equally. That's the most a distinctive character or description, that the atoms in the polar covalent bond, they do not share the electrons equally. Unequal sharing of these electrons causes a partial positive and negative charge for each atoms or a molecule. So these unequal sharings of these electrons causes or can cause a partial a positive or negative charge for each of these molecules. But you have to very look out for the description of a polar and non-polar covalent bond. When we use the use of an example, we talk about the hydrogen, where we talk about the water, sorry, H2O, where you see there are 2 hydrogen and 1 oxygen. And I did mention about ionic bonds when I started this, when we're starting talk about the bonds. Atoms sometimes strip electrons from their bonding partners, not from themselves, but from their bonding partners. For example, the transfer of an electron from sodium to chlorine, where you're gonna have to have sodium chlorine as an output or as a result. After the transfer of that electron, both atoms have now charges. They have charges. Not now, but they have charges. A charged atom or a molecule is called an ion. What is an ion? It's a charged atom or a charged molecule. Simple as that. So where then if there's a transfer of electrons from one element to another, those transfer of an electron, both atoms have become, or they will have within them charges. Then this is an example where sodium is going to be transferred into chlorine, where you're going to have a double salt of an sodium chlorine. And here also they put also the ionic bonds, where you're going to see sodium, where you're going to have 11 protons and 11 electrons and protons. Here in chlorine, you see there's also 17 protons and 17 electrons. We're going to come to that, how you can be able to determine that. But you can see here electrons 2, 4, 6, 8, 10, 12, 14, 16, and 17. If my calculation, then here is 2, 4, 6, 8, 10, and 11. Then there's what we call a cation, which is a positively charged ion, and anion, not onion, anion, forgive my pronunciation, which is negatively charged ion. And ionic bond is an attraction between an anion and a cation. So ionic bond is an attraction between a positive charge ion and a negative charge ion. in case they put it in a different perspective or in a different format, you might have to know it and get it in that format. So a calcium is a positively charged ion, and anion is a negatively charged ion. So ionic bond is an attraction between the two, between the positively charged ion and the negatively charged ion. Compounds formed by ionic bonds are called ionic compounds or salt, but I'll prefer you go with ionic compounds because if they wanna, I've seen most of ionic compounds on the previous question paper than salts. So salts such as sodium, chlorine, table salts are often found in nature, such as crystals. We know that, and then this is just the description and the diagram which is showing that. So with chemical interaction, as I conclude, most of the strong bonds in organism are covalent bonds that form cells, molecules, and many of those large biological molecules are held in their functional by weak bonds, and the reversibility of weak bonds can be advantageous. And then another bonds which are there, there's hydrogen bond. We're talking about ionic bond. Hydrogen bonds are forms when a hydrogen atom covalently bonded to one electronegative atom is also attracted to another electronegative atom. So these ones are very weird. They are formed when their atom, which are covalently bonded, they are bonded to one electronegative atom and also attracted to another electronegative atom. So, for example, in living cells, the electronegative partners are usually oxygen and nitrogen. That's where you're gonna see the kind of example of hydrogen bonds, where you're gonna find them, which are they are attracted or they're covalently bond to one other electronegative atoms and the other. Then van der Waals interactions. If electrons are not evenly distributed, they may accumulate by chance in one part of a molecule. So this is the eventuality that can happen if these electrons are not evenly distributed, where we're going to have to have these van der Waals interactions, which are interactions between molecules that are close together as a result of these changes. What these changes when these electrons are not evenly distributed, where they could result in the accumulation of other by chance with one part of the molecule. Collectively, such interaction can be strong as between molecules of glucose powerhead. This is just an example, don't worry about this, and the wall of a surface, and then there could be that. Any question? I'm going to end here today.